words. My name is Wilhelm Huck, and I'm your host tonight. In the past 5 weeks, 5 months, I should say, we have been exploring all kinds of artificial life, tissues that are grown in the lab and that resemble natural tissues, tissues that can actually resemble robots and essentially robots made out of cells. We have seen robots that interact with human beings. We have seen sort of attempts to make a synthetic cell, all kinds of examples that you might argue are forms of life. But the biggest question that we haven't asked at all is, what is life in the first place? And of course, it could be that we all say, well, life is something that we recognize when we see it, and that is perfectly acceptable for many of us, and it's a good working definition. But today's speaker will certainly not be happy with a definition like that. Today's speaker is Professor Carol Cleland from the University of Colorado, Boulder. She's a philosopher. She wasn't trained as a philosopher originally. She was a mathematician and then a software engineer before she moved to philosophy. And she's interested in the philosophy of science, the sort of bigger questions and the really big questions. And she was one of the people who coined the term of a sort of a separate biosphere, where you could say, well, maybe there is a form of life on Earth that we simply wouldn't, this is the shadow biosphere, by the way, a form of life that we wouldn't recognize as such because it wouldn't operate on the same principles, but it would still be a life. So I couldn't imagine having a better speaker at the very end of our second series of the late night conference as Professor Carol Cleland. And I would like to hand the Zoom over to her. As usual, after the lecture, please feel free to ask questions and start a discussion. Also, if you are in the YouTube, put your questions in the chat and we will read them out over here. And so with that, Professor Cleland, I move over to you.
Thank you. Thank you for inviting me to participate in this. I'm looking forward to it. So part of the issue here is what is life? And I'm going to talk about how you can answer that question, but I'm going to be very skeptical about whether or not we're in a position to provide the kind of answer to the question that most of you would expect me to supply, like a definition or a theory of life. And I'm going to explain why I'm so skeptical about our being able to answer that question now with the resources we currently have, and tell you what we need in order to be able to answer that question. So, whoops, let me see if it's not moving there. So there are really two popular strategies for answering the question, what is life? One is what was mentioned by the host of this session, define life. That's the most popular. Everybody wants to come up with a definition of life. And the other one is appeal to a preliminary, truly general, that is universal theory of life. And I'm going to tell you why neither of these approaches is going to be satisfactory. That's the first part of this talk. And the second part of the talk, I'm going to tell you, I'm not going to leave you saying, well, what do we do? How will we recognize an extraterrestrial form of life if we had encountered it, if we don't have a definition or a theory of life? And I'm going to tell you a way of coming around the problem of how to do this if you lack a definition or theory. So the bad news of these two is neither is up to the task. So I'm going to first start with some logical and philosophical problems with defining life. Definitions are concerned with language and concepts. After all, when you want the definition of a word, you look it up in a dictionary, right? And it gives you another set of words which have the same meaning. So the point is, definitions are about language and concepts. They can't go beyond our current understanding of life and tell us about things in a mind-independent world of nature. The most informative definitions, which I'm going to dub for simplicity of discussion, ideal definitions, these are the ones that logicians and philosophers love, specify the meaning or conceptual content of linguistic expressions. That's why you look them up in a dictionary, by supplying necessary and sufficient conditions for the application of the term being defined. Now, dictionary definitions don't do this. This is what logicians do. So if you want to actually know the meaning of a term like bachelor, what you do is you try to come up with a synonym for bachelor. That is to say, a group of words whose concept is going to capture what it means to be a bachelor. And if you really have a very informative definition, what that set of words that define the word bachelor is going to do is provide necessary and sufficient conditions. Anything that's a bachelor is going to be an unmarried human male if the definition is truly ideal, and anything that's a bachelor is going to be an unmarried human male, and vice versa. Note that the single quotes around bachelor, that indicates that we're not talking about bachelors per se. We're specifying what a word, bachelor, that's the single quotes here, means in English. And that's the way in which the best kinds of definitions work, which aren't the ones found in dictionaries. The best kind, the ones in dictionaries are usually circular. And I don't want to go into detail about dictionary definitions, but if you're a logician or a philosopher, you want to do better than that. And anybody who's taken an elementary informal logic course has been subjected to this. So the idea here is a bachelor means unmarried human male, doesn't talk about bachelors per se. Now you might say, well, that's not a very good definition. What about a baby? We wouldn't call a baby a bachelor, it's an unmarried human male. Well, we can add adult to fix that up. That's what you do in informal logic courses. And then you have to worry about the Pope. And the lesson here is that language is vague. So we come as close as we can to an ideal definition, supplying necessary and sufficient conditions, but we can't get it perfectly right, because then our language would never be able to be applied to new situations. So language, the vagueness of language is actually an advantage to language. It allows us to apply words to new situations, because if you have necessary and sufficient conditions, you're going to run into the problem that you completely boxed up the meaning of the term. And language doesn't work that way. So ideal definitions work fairly well, but there is a problem of vagueness for answering what is questions about what we'll call artificial categories, that is human categories. Bachelor, garbage, chair are examples of human categories, categories that are based on human interests and concerns. So garbage, what we call garbage, depends on human interests and concerns. Similarly, bachelor depends on human interests and concerns. You don't find bachelors in nature or garbage in nature in the sense that you do when you're talking about human society and culture. But certainly the best answer to the question, what is a bachelor, is going to be unmarried adult human male. Yeah, the Pope's a problem, but he's not a big problem here. But where you get a very different situation is when you try to come up with an ideal definition for a what is question about a natural category, such as water, temperature, lightning, etc. These words are really different than the words that we had earlier, bachelor, garbage, and chair. How are they different? Well, there wouldn't be bachelors or garbage or chairs if there hadn't been any humans. There would be water, temperature, and lightning. Temperature and lightning are phenomena in a mind-independent nature. And so the problem is that ideal definitions don't work very well for them. I can explain exactly why that is the case. Consider the answer to what is water. The answer is going to be H2O. But no amount of dissecting the concept of water is going to tell you that. Whereas dissecting the concept of bachelor, if somebody said, oh, I've just discovered that bachelors are, unmarried adult human females can also be bachelors, you would say you don't understand what the word bachelor means in English. But if somebody says, just discovered that water is composed of H2O, you're not going to say that. Even if you're talking about at the 17th century, people didn't know, had no concept of the idea of hydrogen and oxygen in the periodic table, they would have come up something different. They would have come up attempts to answer the question by paying attention to the perceivable properties of water. They don't have a periodic table. They don't have constant hydrogen and oxygen. And so how would a 17th century scientist answer the question what water by defining water? Well, knowledge, frankly, is limited. He, in those days, women scientists were, but they weren't taken seriously, and they didn't get the credit they should have. Knowledge of water limited to properties of water to be perceived, be wet, transparent, tasteless, and a good solvent. Not to concepts like hydrogen, oxygen, and the periodic table and molecular bonding. None of that is available to our 17th century scientists. Many of the substances identified by our scientists' contemporaries lack one or more of these properties. Muddy water is not transparent. Brackish water is not odorless. So the question is, if our 17th century scientist wants to define water, what property should he choose? And it's certainly not going to be H2O. So I love this example. Here's Leonardo da Vinci, who was, as well as being a great artist, was also a scientist. He was very interested in water. In fact, here you could see the utter complexity that da Vinci faces when trying to figure out what water is. I love this quote because it really shows you. Now you have to remember, he doesn't have the periodic table, doesn't have concepts of oxygen and hydrogen. So he's limited to the sensible properties of water. And this quote expresses his utter perplexity about which ones to pick and focus on. Water is sometimes sharp and sometimes strong, sometimes acid, sometimes bitter, sometimes thick, and et cetera, et cetera, et cetera. Now we, in the modern times, can explain these features by saying, oh, it's not pure H2O. It has various other kinds of chemicals in it. That's not available to da Vinci. And he stuck sensible properties of water. And the sensible properties of water, call water, vary dramatically in different contexts. So it's not like the Bachelor case. If he analyzes his concept of water, he's stuck with the sensible properties of water. So the alchemists, which were chemists, they did a lot of important experimental work. And that experimental work was actually very important in the 19th century when people began constructing the periodic table and concepts of molecular bonding, et cetera, because they could go back and look at the various research. They discovered a lot of things, how you combine things, what chemicals you get. They just had the wrong theory to understand it. And so the alchemists chose solvency as the essential property of water and classified nitric acid, mixtures of hydrochloric acid, which are even better solvent than water, as water. It's not an accident that they called it agua fortis, that is nitric acid, and mixtures of hydrochloric acid, agua regia. Agua means water. And the regia is interesting because hydrochloric acid can dissolve gold, so it was royal. And agua fortis was, strong water. Nitric acid is very strong. It's not an accident that they called them aqua fortis, agua regia, because for them, they had, if they had water as being a good solvent, they might have, if they were interested in definitions, defined water as a good solvent. But we know they're wrong. These chemical substances are different molecular compounds. Water is H2O, nitric acid is HNO3. They couldn't have known that because they didn't have the concept of a periodic table of nitrogen, hydrogen, and oxygen and molecular bond. So they didn't have access to any of this. What happened was people discovered later that stuff we call water is composed of these molecular compounds. And the important point here is no amount of analysis of the perceivable properties of water familiar to a 17th century chemist could have revealed that water is H2O or nitric acid is HNO3. And that shows you a really big difference between natural kind terms like lightning and water and artificial kind terms like bachelor and garbage. So the point is, you can't really define water. And now I'm going to go and the same is true of life, insofar as life is a natural kind term, that there would be life even if there had never been any humans, because they are about a mind independent reality. In the days of the dinosaurs, when there weren't any humans, I presume there was still life, just as there was still water. But there weren't any bachelors, and there wasn't any garbage, and there weren't any chairs, because the concept of a chair and garbage and water is a human concept, dependent upon our social structure. So you're defining life is a no starter because of the very nature of definitions. So what about a theory of life? That would be the kind of thing that we would hope would work, a tentative theory of life. It turns out that you just can't base an adequately general theory of life on life as we know it on Earth. And I'm going to explain why not now. So you might think the amazing morphological, that's the shapes and structure, diversity of Earth life suggests that all the possibilities for life are represented on Earth today. Look at all these different forms of life. There's a bacterium. Look at all these things, mushrooms, snakes, elephants, trees, birds. I could have gone on and on. The variety is enormous. And so you might think all the possibilities for life in the universe are represented if you had a concept of life that was based on morphology, which, by the way, people did prior to Darwin. They had a morphological notion of life. But it turns out that a morphological account of life, the morphological diversity of Earth life, is deceiving. Why? Well, we know a lot more than did the pre-Darwinians about life. We even know more than Darwin did about life. All known life on Earth, from bacterium to trees to elephant, all of these things here are actually remarkably similar at the molecular level. I'm going to talk about that in a minute. But on the basis of this molecular similarity, which we'll talk about in a minute, we have found that all life on Earth shares a microbial last universal common ancestor, affectionately known as LUCA, and hence represents a single example of life. Looking at the genetic genomes of everything from bacteria, all of these things here, we discover we can construct this tree, which bottoms out in what must be either a single organism or a community of organisms called, we said, the last universal common ancestor. And here you are. Look at animals. They amount to much. And ironically, most of the stuff I showed you here was animals. The greatest diversity is among the single-celled prokaryotes, bacteria, archaea, and eukaryotic celled organisms. And so complex multicellular organisms are a tiny part of life as we know it on Earth today. But the important lesson from this phylogenetic tree of life, constructed looking at genetic sequences of all of these organisms and their genes, is that all life on Earth represents a single example of life. Moreover, known life on Earth, remember I say known because I happen to think there might be a biosphere, but we're going to ignore that for the moment because we don't know that. Known life on Earth is astonishingly similar at the molecular level, and most importantly, could have differed at least modestly from familiar life at the molecular level. And we don't know how different it could have been. So I'm going to talk about the biomolecules, two of them, proteins and nucleic acids, to make this clear about how similar all life is, from the bacteria to a redwood tree to is. And this is the evidence on which the hypothesis of LUCA is based. So proteins supply the bulk of the structural and catalytic, that is enzymatic material, familiar life. What basically builds your bodies and powers them. That's what catalysis does. And proteins are made up of long strings of something called amino acids, just long chains of amino acids. And here is a 3 amino acid chain. It would be peptides, small pieces of a chain of amino acids are known as peptides. So this is small enough to be a peptide. Actual proteins are usually really long. So what you can see here is that all amino acids are remarkably the same. Here's an amino acid, here's an amino acid, and here's an amino acid. And you'll notice that all of this stuff is remarkably similar. And they're all bound together by these things called peptide bonds. The way in which amino acids differ is just in the position of these R's. Long chains of these amino acids make up the proteins, which supply the structure of your body and the chemicals that power it. All known life, from a bacterium to uses the same 20 amino acids. Yet over 100 amino acids are found in nature. Now that's pretty startling. Think about it. A bacterium and you all use basically the same 20 amino acids, but there are over 100 amino acids found in nature that aren't used in biology, and they're supplied meteorites, and they're supplied by chemical reactions and thermal events create these amino acids. But life on Earth only uses 20 of them. That's quite amazing. Biochemists, this is more amazing, have synthesized non-standard proteins from abiotic amino acids that are not used by familiar life, and that would be functional in the right organismal environment, but it doesn't exist on Earth. So that's kind of shocking that all life is so similar in the structure of its proteins. Same is true of its nucleic acids. Nucleic acid store and manage the hereditary information of familiar life. So you're all familiar with DNA, the double helix. Here's what we've got here. RNA involves the transfer of information from DNA to ribosomes, which construct proteins. All known life on Earth uses the same nucleobases. Here they are, there are 4 here. It turns out that in RNA, T is uracil is substituted for, which is, we'll abbreviate as T for thymine. So there are actually 5 bases, but depending on the molecule, only 4 are used. 4 are used in DNA and 4 are used in RNA. There's just a switch of U for T in RNA. And the same sugar phosphate bond backbone. Here's the sugar phosphate backbone. All known life on Earth uses the same triplet genetic code and coding scheme. So this is pretty amazing. So the code for the amino acids that is stored in DNA and then replicated in RNA and taken to a ribosome to construct a protein is a triplet code. Here we see it. And here is the first base position, the second base position, and then the third base position. And you can see then you have UUU, UUC, UUA, UUG. These basically, so UUU and UUC both code for the same amino acid. And the same here, UUA and UUG code for the same amino acid. So you have redundancy in the code, but you don't have, you don't have the same code coding for 2 different amino acids. So there is it's kind of many to one, but it's not many to many. So that's really important here. And this redundancy then has allowed biochemists, to create non-standard nucleic acids and to code for non-standard proteins. So for example, Steve Binner has been somebody who's done a lot of this work. What you can do then, and you can see this here, is you can basically take, and here's what's happened here, you can take these redundancies in the code and have add a new code and have them, or just add a new code and have them code for a different, amino acid. And that's what we see here. Here's the standard genetic code. Here's the expanded expanded new genetic code where we add a new nucleo base and not a nucleo. We add a new basically coding scheme, and then we code for a different amino acid. So we've constructed here a non-standard protein that doesn't exist for any sort of known natural life on Earth. And we have a non-standard genetic code with different nucleobases. He's done all of that. And so we know that life could have been at least modestly different than it is in fact on Earth. And we don't know how different it could have been. And that brings us up to what I'm going to call the very serious N=1 problem of biology. One can't generalize to all life, wherever it may be found in the universe, from a single example that we have reason to believe could be unrepresentative life. What's our reason? This is our reason. We know that life could have been modestly different in terms of its biomolecules, both proteins and nucleic acids and genetic code. But we don't know how different it could have been. So we know it could have been modestly unrepresentative. We don't know how unrepresentative. We don't know whether you could have had different informational molecules. Some people have suggested alternative information molecules to DNA. We just don't know this. And so we really are stuck with this tremendous problem of dealing with a single example of life, which may be unrepresentative life. And it's just a logical problem. You can't generalize on the basis of a single example like that. And so let me give you an example, the zebra analogy. Supposing supposing aliens came down and they wanted to come up with the concept of a concept of mammals. And all they have available to analyze are zebras. So they want to understand what a mammal is. And the only examples they have of mammals are zebras. What do you think they're going to generalize on the basis of? Their stripes, which are very ubiquitous among zebras and quite distinctive, as you can see, or their mammary glands, which only half of zebras have, the females. You can bet they're not going to go for the mammary glands because they're going to think that, well, those aren't universal. Yet we now know that the mammary glands of mammals are more important to their identity as mammals than certainly stripes, which many mammals don't have. So that illustrates the problem. We don't know whether a single example of life is like the zebra with regard to mammals. We just don't know that. And the problem is we're going to be looking for universal features of our single example. And maybe the features we're focusing on are not the right features. We just don't know that. So we aren't in a position to come up with universal theory of life. So this leads us to a dilemma. In order to answer the question, what is life? We need additional examples of life. But how can we recognize truly novel forms of life, examples of life as we don't know it, if we don't have a definition or universal theory of life? It's not at all obvious. Biologists are always saying we'll recognize it if we see it, but that's not at all obvious. You'll recognize it only if it's sufficiently similar to familiar life. And it might not be, especially if it's microbial. So here's the suggestion. Searching for life with either a definition or tentative theory of life based too closely on familiar Earth life runs the risk of blinding us to truly novel forms of life. We'll recognize alien forms of life only if they resemble Earth life in designated ways. It's a little analogous to looking for your keys under a searchlight, because the theory or definition provides you with a searchlight, but you don't know what's out here in the dark that you haven't really considered because it's just too different from familiar life. So my solution is use tentative versus defining criteria to search for anomalies. Instead of searching for familiar life via an Earth-centric definition or theory of life, search for anomalies. Anomalies are physical systems that both resemble and fail to resemble life as we know it in provocative and unanticipated ways. This contrasts with the definitional approach, which identifies cases that do not conform to the favored definition as non-living, period. So the idea here is that you want to look for physical systems that shouldn't be there. That is to say that you say, like, what? This doesn't seem like life, but it has certain properties that are very suggestive of life. but it also has properties that are suggestive of non-life. These are the kind of systems that are going to straddle the boundary of any Earth-based definition or Earth-based tentative theory. And they're the ones that are going to allow us to expand our concept of life beyond its Earth-centric boundaries, which is all that we have accessible. Once identified, an anomalous physical system becomes a focus for further scientific research, which contrasts with the definitional approach, which identifies cases that do not conform to the favored definition as non-living, period. And now I have an example I'd like to develop. I need another 10 minutes. Can I have that? I'm going to go ahead then. I haven't, nobody said no, but I know you. Okay. So an example of a potentially biological anomaly. So I've just talked about anomalies. Now I want to give you an example to fill in this concept of what a potentially biological anomaly is. And that's what I think NASA should be looking for. Not going with a definition or tentative theory, but looking for potentially biological anomalies. That is systems, physical systems that are provocatively similar to life in some ways, but provocatively different in other ways. So I think we've actually run into that. And I think the response tells us exactly why definitions are inadequate. The mysterious Martian oxidant discovered during the Viking missions to Mars in the 1970s are going to be the topic here. So the Viking biology experiments of the 1970s consisted of 3 metabolic experiments based on an operationalized metabolic definition of life closely based on Earth life. So the basic idea here was that you had, so I'm going to talk actually about one of the experiments, which all 3, by the way, initially showed positive results, but there's one called the labeled release experiment, which to this day remains deeply puzzling. And that's the one I'm going to focus on here. So basically the labeled release experiment worked like this. You had a broth of nutrients which were radioactively labeled with carbon-14. And the reason you did this is you wanted to supply the broth of nutrients to the Martian soil and then see whether or not there were little Martians in there that would metabolize it and release radioactively labeled carbon dioxide gas. This happens with Earth microbes. And they had taken, the broth was constructed from all the different nutrients that organisms, microorganisms on Earth had been shown to be able to metabolize. So it was this broad sequence of nutrients, basically amino acids. Remember, that's the composition of proteins. It was basically amino acids that known cultivatable Earth life could digest. And then the idea was, and it was, the carbon in that was radioactive, so that when they added the nutrients to Martian soil, which they did, they had a radioactivity detector, which would detect whether that particular nutrient solution was metabolized, because if it was metabolized, it would release radioactively labeled carbon dioxide gas. And in fact, that's what it did. That's what they got. They got radioactively released carbon dioxide gas. And this was very exciting. And I'm going to now tell you why NASA concluded no, the Vikings didn't find Martian microbes. They concluded no because the response, the release of this radioactively labeled gas happened faster than compared to Earth microbes. They had run this experiment with Earth microbes, and basically Earth microbes were slower in releasing carbon dioxide. Their metabolic process was slower than the results that they got with the labeled release experiments from the Martian soil. A second injection of labeled release nutrients onto positive soil samples, that is to say, samples that had already released radioactively labeled gas, you would expect it to be release additional radioactive gas, because the hungry microbes, the microbes would now be hungry, the Martian microbes, and they would now metabolize the new nutrients they've been given. That didn't happen. That did happen when they ran the test. They tested it on Earth. That's exactly what happened. When you gave the first injection, they released radioactive, this is Earth microbes, gas, labeled gas. And then when you waited a little while and you gave them a second helping, it released more radioactively labeled gas. That's what they expected to find. They didn't get that. And in fact, what happened was about 30% of the gas that was released after the first injection was bafflingly reabsorbed into the soil. They've never seen anything like that. It was absolutely baffling and not at all what they expect from Earth microbes. So they actually were so deeply puzzled because they couldn't explain this. It turns out that not only they had two basically experiments. They had this one where they had the release of the gas. Whoops, sorry. This one where they had the release of the gas. And then they actually took a sample of soil and they sterilized it at high temperatures, and they didn't get the release of the gas when the nutrients were added to the soil, which was exactly what you expect of Earth microbes, because you would have killed the microbes. So they got those two results, but NASA still said no, because it wasn't quite like it was on Earth. And then you got this baffling result. So they then relied on an instrument, the gas chromatograph mass spectrometer, which hadn't been intended as a light detection instrument, but it ended up being one because basically they found no organic compounds in the Martian soil. Organic compounds means, carbon compounds like we have, life is composed of on Earth. So they concluded no. But the PIs, principal investigators of the labeled release experiment, said NASA is wrong. They concluded yes. Why? Because the Martian soil sample did just what NASA's sanctioned metabolic definition stipulated. The soil released radioactively labeled carbon dioxide gas when inoculated with the radioactively labeled nutrient solution. When heated to a temperature of 160 degrees for 3 hours, a temperature high enough to kill any known bacteria, no more radioactively labeled gas was released by the soil sample. This is exactly what you get with Earth microbes. So they said yes. Here's a quote from Gil Levins. Quote, the Viking mission labeled release data detection experiment returned data from Mars fully satisfying the pre-mission, think defining, metabolic definition. That was what they were working with, criteria for the detection of microbial metabolism. But NASA said no, and NASA scientists began trying to conjecture about biological candidates for the mysterious Martian oxidant that could explain why you got the results that were extolled by Gil Levins and his team. Namely, it appeared it was very lifelike in terms of the release of the gas, and then when you sterilize the soil, the lack of release of the gas. And so they tried to come up with an account in terms of non-biological chemicals that could explain the results of this experiment. And they explored over the years many different ones: hydrogen peroxide, superoxides, exotic oxidation states of iron. None of them worked. The most popular one recently is perchlorate, which was accidentally discovered by the Phoenix lander. And it turns out it's a very reactive gas. So they said, gee, this is probably it's a good oxidant. And so this is probably what it is, explains what happened in the LR experiment. The problem is it doesn't. Why? Because perchlorate only becomes reactive above 200 degrees Celsius. And the temperatures in the labeled release experiment container were no higher than 10 degrees Celsius. And so you could explain by perchlorate why the GCMS, gas chromatograph mass spectrometer, found no organic molecules in the Martian soil, because it heated them up to 500 degrees. Plenty high for perchlorate to become reactive and to have basically killed or any of the organic material, amino acids or whatever in the soil. So you could explain why the GCMS failed to find any organic molecules, but you couldn't explain the results of the LR experiment itself. And yet, ironically, perchlorate is still being extolled as the solution. And when people point out what I just point out to you, people say, well, maybe there are these exotic reactions that can make perchlorate more reactive at lower temperatures. Maybe it breaks down under ultraviolet light or under radiation to more complex and more highly reactive at lower temperatures compounds. This is all highly speculative. But what's really interesting is life is also an oxidant. Yet NASA has never included an instrument package to identify this mysterious Martian oxidant. Why? Because their official mission was to find life, and they concluded that the cause of the mysterious chemical reaction in the LR experiment wasn't life. Why? Because it wasn't enough like Earth life. And this shows you the danger of going with the definition. They did go with the metabolic definition. NASA has now moved on to the chemical Darwinian definition, but the same arguments I'm giving here are going to hold for it. And it's even harder to test because you have to look for biological evolution, and that's a time, takes a while to occur. So this is a classic example why definitions are problematic. The truth is neither side of the debate or the LR experiment knows what they found. The results of the LR experiment were potentially biological anomalous. They were anomalous in the sense that there were striking features that seemed biological. That's why Gil Livens and his team said they found life. But there were also features that seemed like they weren't biological, like the reabsorption of the gas and the failure to reinvigorate when a second sample was added to the second nutrient solution was added to the previous basically the soil that had previously released radioactively labeled carbon dioxide gas. And had they been treated as such, anomalous, both possibilities, biotic and abiotic, would have been seriously entertained and explored, but they didn't. And that's an example of why you should be searching for anomalies and not going with the definition or tentative theory of life. I think that we have a potentially biological anomaly in the LR experiment, and NASA needs to go back and look for it. But I talked once to a very famous, very senior NASA scientist and said, why haven't you done that? Turns out they did have a mission to do that, but the mission got scrubbed. I can't remember what happened, but it didn't. But they never followed up on it. Why? Because as this scientist said, well, we're looking for life, we didn't find it. I'm not convinced that they didn't find it. I think they might have found it, but it wasn't like our form of life. Thank you. Shall I stop sharing or leave it up?
Carol, thank you for a very thought-provoking and fascinating talk. I thought when you were going to say in the beginning that you are not going to answer the question of what is life, and you explained that by looking at different life forms and trying to define it, that will not work, and having a theory that will not work. And I thought, okay, what is going to happen? And I think you really put us into different directions, I think, to start thinking about different ways of thinking about that problem. So thank you so much for that. I had one question, though, that I wanted to ask you first, because you said, well, we know only one example of life, essentially, because you say, well, all life derives back to, or can be traced back to Luca, and so the essential biochemistry almost, or the central dogma, is the same in all forms of life. What now if it turns out that life is so unique that you could only have one functional form of life? Is that something that you would consider, or is that something that you would?
Yes, I mean, I think all options are open here. We just don't know. We do know that it could have been modestly different. Keep in mind the kind of work that Steve Binner and other synthetic biologists have done. They have created microbes with actually a different genetic code, different nucleobases for storing genetic information, and constructing non-standard proteins. So we do know life could have been modestly different. So it's not that we have, and that's the problem. We don't know how different, because all of the work of people like Steve Binner have been top down. That is to say, they're using our form of life to construct modest alternatives, but they can't go beyond that because they don't know what a non-standard information molecule that wasn't like DNA would even look like.
I have one question here already on the screen, and then we move to the audience. And first question in the
Shall
audience.
I get rid of the picture so I can look at people, or do you want me to keep up the?
Yeah, I think we can do that if people can switch the sort of one of the panels to the sort of audience up. Well, while we're doing that, I wanted to ask a question is, what is the role, Will Robinson is asking, what is the role of the context in the definition of life? For example, could a distinguishable living system in a relatively simple environment be indistinguishable in another more complex environment?
So I don't know the answer to the question, what is life? What I want to say is that we want to look for, a system that, and all environments are complex. I just don't believe in such a thing as a simple environment, because the chemistry of any environment that's a natural environment is going to be complex. Now you can make them simple in a laboratory, but you can't really do it. The Martian environment may seem very lacking in complexity compared to our environment on Earth, on a rainforest. But it's actually fairly complex if you ask astrobiologists, because, there's all kinds of interesting chemistry that's going on, and it's very dry, and who knows what's slightly beneath the surface. So what I will say is that I just think the whole idea of trying to define life is going to lock you into a concept of life that will blind you to genuinely alternative forms of life. I don't know whether that answers your question.
No, I think I understand. I definitely understand your answer there. There's a question right here in the front.
How does this work? Oh,
Still
like this,
working, yeah.
right. Yeah, thanks for the talk. I was surprised actually, because the definition of life that I learned at biology class at school is actually very different from the definition you were giving, and I think much broader. So I learned that something is alive if it can use nutrients to grow, if it can sense the environment and react to it, and if it can reproduce itself and pass on the code on its own. And I could also name a few anomalies that I've discussed with other life scientists. Some of them are very known, viruses and prions. And I've even had the thought experiment with a few colleagues about fire, whether it would be alive or not. But could you comment on that definition? And would that definition, which is not bound to molecules, work better?
No, I don't think it does, because if you try, so you've just given a combination of a metabolic definition, that was the first part of what you described, and a genetic definition, reproducing and then responding to the environment. Now, if you're going to make that very specific to our form of life, it's going to be too specific. But let's consider a hurricane. A hurricane extracts energy from its environment to maintain itself for fairly remarkably, many of them, long period of time. You can think of a tornado too, but hurricanes I like because they're longer, they take longer. And so a hurricane is going to last longer and be metabolic in this general sense of extracting material from the environment to maintain self-organization than a mayfly, which lasts a single day. Now, in terms of reproduction, you have to think about this, about whether or not you want to say that any organism that doesn't reproduce isn't alive. And you're going to immediately run into problems with sterile organisms and hybrid organisms like mules that can't reproduce. Are you going to say they're not alive because they can't reproduce? I don't think so. And so I think that you can easily pick apart that definition, which is one that, elementary school kids are given, but I don't think many scientists would accept those. And so the attempts like metabolic definitions that NASA uses was using is an attempt to sort of avoid some of these problems. Any definition that you have that you're going to use to search for life has to be become more specific. And so you'll notice the metabolic definition that the labeled release experiment used wasn't a general metabolic definition. It was very specific. It was based on, a nutrient solution of a certain kind and the sort of metabolism of that nutrient solution that would occur that would be like Earth organisms. You can imagine all kinds of different kinds of metabolism that might not release CO2 gas, right? We release oxygen, for example, in our metabolic processes. So I think those simple-minded definitions are meant for young people to sort of think about more general properties of Earth life.
Carol, I had another question because I know you were associated with the NASA astrobiology activities and also with the SETI Institute. And I thought if NASA spends all that money to go to Mars to find life, then what do you think they should really be looking for? Because they're not going to find living, or probably not finding living bacteria or something. Would they be looking for signatures of life?
Yes, of course, to be looking for signatures, because looking for microbes on another planet is going to be very difficult. You're going to have to look for the kinds of things that the microbes do. You'll be looking for things like maybe structures, like stromatolite structures, which are hard to understand forming geologically because of the angles, and the little microbes will basically secrete material that glues on grains of sand. So structures like that are very difficult to explain abiologically are a good thing to look at. But you can't rule out finding weird abiological processes in other worlds too. We've actually done that. We found there's plate tectonics on Pluto with ice. That's pretty astonishing. Nobody expected that. So as we go out to other worlds, we're going to find weird stuff that's not just living. And there is this kind of assumption if we find anything weird, it's going to be living, but we're going to understand all the non-biological processes. And that's just not true. And there are a lot of puzzles I could tell you about Mars that nobody understands. That are almost certainly abiological. What we want to look for are, so rather than going with a definition, like a metabolic or a genetic definition, my view is, take the universal features of life and include them, but also include features that are specific to particular environments on Earth, which represent adaptations. Take a set of characteristics that are designed for a particular environment and include the things that we think are probably might be universal to life, but don't be wedded to it like a definition. Look for things that have some of these features. Maybe you get something that looks like metabolism, but nothing that looks like a genome, but you have some other weird features. And instead of concluding, as NASA did in the case of the LR experiment, oh, well, it's not enough like our life, it's not life. What you do is you say, hey, this is really provocative. We don't understand it. It is potentially biological, but it might not be. And now focus your research on those kinds, the most provocative of those systems. Does that answer your question?
I think it does. There's a question right on the top.
Yeah. Hi, Carol. Thanks a lot for your awesome talk. I was curious what you thought about the idea of evolution in terms of life and how we should define it. Do you think this is a feature that would be applicable to n=2, n=3, beyond Earth scenarios?
I really like your question because I have real doubts about that. And let me tell you why. I think that the Earth is in a kind of Goldilocks place for evolutionary processes. But if you had an environment that was very stable, which Earth isn't. Earth isn't too fast and it's not too slow. If it's too fast, you get extinctions. If it's too slow, you are going to get different kinds of processes. So I can imagine an environment that doesn't change very much. And I think that some microbial environments deep down in the Earth are probably like this. And you would find that in such an environment, you might have Darwinian evolution be maladaptive, because chance mutations would produce organisms that are maladapted to the environment, since it's already well adapted. Where Darwinian evolution works really great is an environment that's changing, but not too fast, because then these modest mutations are going to give you a supply of material so that you can adapt to changes in the environment. I think it's really interesting that microbes have a mechanism called lateral gene transfer for more quickly changing their genetics than creatures that are multicellular eukaryotes, which depend upon, on lateral passing, I'm sorry, not lateral, but passing genes from the parents to offspring. That's a slower process, but microbes can exchange genes directly. And so one microbe can now affect the survival of another existing microbe without having to wait until you have offspring. And waiting until you have offspring is more, shall we say, fraught, because why? Because you might die before you have your offspring. But if something can actually give you an adaptive gene, which is what happens in lateral gene transfer, then you can survive the process. And you'll notice that from probably microbes go back to, I mean, some people are pushing it to 4.1 billion years, evidence of microbial life. And you don't see any evidence of complex multicellular life until basically less than 1 billion years ago. And the earlier Earth was not as, shall we say, stable as the current environment has been. I mean, you were hit by meteorites, you had probably the entire oceans evaporated, you had all kinds of ecological changes that were much more rapid. And lateral gene transfer would be much more adaptive in those circumstances. And people keep trying to fit it into the Darwinian account, and it doesn't fit as well as people think. And so I think that's a good example of how you might have different kinds of genetic processes adapted to different kinds of environments.
Well, I'm looking for questions in the audience. Otherwise, I had a question myself. You're looking at sort of approaching the question of what is life by still looking, I think, at a lot of processes or phenomena that we can observe when we observe a whole range of different living systems. And then I think also when we would be looking in extraterrestrial life, I think we would still be looking at something similar in terms of indeed growth or adaptation or sort of multiplication or something, but it doesn't bring us maybe closer to another question that we could ask that might, I think, in my opinion, get us maybe closer to the question of what is life, if we ask ourselves the question of what is the meaning of life. Because maybe there's an inherent property of a living system that makes it actually alive, that is the crucial bit of information that will also help us maybe to understand what life actually really is.
So you sound like a vitalist.
Molecular or maybe molecular or thermodynamic point of view.
So you sounded there like a vitalist. The vitalists used to think there was a special property that all living systems had. Of course, nobody is a vitalist anymore. You could go to a thermodynamic definition of life, but then you have the problem of hurricanes and tornadoes, which do extract energy from. They maintain pockets of negative entropy, right? And so that's what's so interesting about life, is that life is able to build itself and sustain itself by extracting energy from the environment and basically bucking the law of thermodynamics. And it does this in local systems. So if you're going to talk about that, you're going to have to talk about hurricanes, candle flames, and all this other stuff. So it's living systems aren't the only systems that have those characteristics. What's really interesting about living systems is they have this mechanism, these complex parts, right, which extract stuff from the environment and keep it going for a long time. There's a sense in which you are you are a continuous chain through your parents and your grandparents all the way back to that first cell. So one of the odd things about life, and I think people who talk about clades versus species are trying to capture that, is that you're part of a long continuous chain that goes back to whatever that first organism was. And that's kind of a shocking thing to think about. So I think that, I'm not sure, what was the rest of your question? So that's one thing, part of my answer to that. What was the rest of your question?
I was indeed thinking on sort of thinking along sort of properties of thermodynamics.
Oh, right, the meaning of life. Okay, and I don't like talk about the meaning of life because this is an ambiguity in the word meaning. When people talk about the meaning of life, they're talking about what things like the Buddhism, or ethics or moral law. How should I lead? The meaning of life for me might be to pursue these interesting questions, or it might be for somebody else to nurture their grandchildren, or it might be for somebody else to pursue politics. In the United States, I think pursuing politics is a very meaningful thing right now, given our current political situation. So the word meaning there is not the same as the word meaning in when you talk about the meaning of a word. Does that make sense to you? And so what I'm really interested in is not so much meaning in these multiple ways of thinking about it, but on what life is viewed as a natural phenomenon. And that might or might not have anything to do with what I think the meaning of life is.
Thank you. And there's maybe one question from V. We can switch on the microphone.
Hello, thank you for your amazing presentation.
I can't hear, so you'll have to translate it.
Hello, sorry.
Hello,
Yes,
can you
I
hear me?
Yes, I can.
Okay, sorry, technical difficulties. My question would be how much the definition of life changed during the last years since you actually started observing maybe the definition itself?
So I think that NASA has finally, and I like to think I had a lot to do with this, given up on going out with the definition of life, like they did with the metabolic definition, or in the 1990s, they came up with the chemical Darwinian definition. I think NASA's moved beyond that to what they call agnostic biosignatures. The problem with agnostic biosignatures is that most of the literature treats them as a partial definition. So you literally read people saying that agnostic biosignature is a necessary condition for life, but not a sufficient condition. That's not being agnostic. Agnostic means that you're open to alternatives. It doesn't mean that you think that, well, I don't have a sufficient condition, but I do have a necessary condition for life. That's not being agnostic. If you're agnostic about the existence of God, you neither believe nor disbelieve. So if you have agnostic biosignature, truly agnostic, you should be doing what I'm suggesting, looking for anomalies with tentative criteria, but that's not what a lot of people with agnostic biosignatures are doing. And so I think calling them agnostic biosignature is basically, a kind of, shall we say, you're misleading yourself and your audience, because it sounds like you're being neutral, but you're not. Does that answer your question?
I think it did. Thank you, Carol. I think that's all we have time for, afraid. So we would like to thank you again for a wonderful and very stimulating and vigorous discussion afterwards. Thank you so much.
Thank you. And do enjoy that beer for me.
Yes, so I was going to say afterwards we are going for a drink, and I think we wait till you're in Cambridge next year, and then I'm sure we'll try.
Well, hopefully I'll be in Cambridge next year, but that would be really great. All
Great.
right, well, thank you.
Thank
Bye.
you. And I would like to thank everyone for listening today. Also, I want to thank especially all the team who made this possible, that we have a nice stage set up and all the sound and lights, and the live stream works well. So well done, guys. Thank you so much. And that was unfortunately all we have for season 2. I guess stay tuned. There will be a season 3. We don't know yet what it will be, but just be surprised. And we hope to see you next year. Thank you all, and we'll join us for a beer. Thank you.